It's time for the 3-question warrant for Biochem 6. Let's get going. What is the most common cause of each of the following? So first we have hypoparathyroidism. The most common cause is actually due to a thyroidectomy. Next we have metastatic disease to the brain. So remember your mnemonic was lots of bad stuff kills glia. So we know that lung and breast cancer, those like to go just about everywhere. Also remember skin, like melanoma, kidney, like renal cell carcinoma, and then GI tract tumors also metastasize to the brain as well. Next we have most common lysosomal storage disease. That's going to be Gaucher's disease. Most common cause of myocarditis is going to be Coxsackie virus or echovirus. Next, what amniotic fluid measurement is indicative of fetal lung maturity? So this is the lecithin to sphingomyelin ratio of greater than 2.0. There are other measurements that can be useful in determining that fetal lung maturity, but the high yield one for you to remember is that lecithin to sphingomyelin ratio. Next, what substances stimulate the release of norepinephrine from neurons? So calcium is what we're looking for here, and that's the normal trigger for the release of norepinephrine. But also think about other drugs like amphetamines, ephedrine, and tyramine. All right, that's going to be it for our warm-up. Let's get to that lecture now.
I don't want to let this video run long, so I'm going to dispense with a funny little introduction and get straight to the lecture. But I do have a couple of extremely high-yield points to share with you. So to save time, I'm going to give them to you telepathically. So get ready. Got that? Great, let's move on. In this video, we're going to discuss the various kinds of lab techniques used to study gene sequences. And then be sure to stick around for the very end of the video after the quiz, because I'm going to go over some tips and strategies for test day. So our first lab technique is called the polymerase chain reaction, or PCR. Now, if you want to study a sequence of DNA, a certain allele or whatever, you need more than one copy of that DNA. You can't do much with just one little strand of DNA. You need millions of copies. So PCR is a way to amplify a specific sequence of DNA. There are 3 basic steps. The first step is denaturation, where you heat the DNA so that the 2 strands separate, and you end up with 2 single strands of DNA. Then the second step is annealing. You cool the DNA, and while you're cooling it, you add some pre-made DNA primers that anneal or bind to the specific sequence to be amplified. Now, these primers give the DNA polymerases a place to start. Remember, DNA polymerases don't start from scratch. They need primers. Under normal in vivo conditions, they have an RNA primer. And then the third step is elongation, where the DNA polymerase starts to replicate the DNA sequence following each primer. So then you would replicate that entire fragment of DNA so that you go from 2 single strands of DNA to 2 double strands of DNA. Then you go through that same process again and again and again, and you get a lot of copies of that one sequence of DNA that you can then analyze. Once you've amplified the DNA sequence with PCR, one of the things you can do to analyze the DNA is called gel electrophoresis. So we use gel electrophoresis to sort and separate PCR products according to size. I'm sure you've seen this before, but on gel electrophoresis, you have a tray filled with this gel. And in one end of the gel, you have little divots, which are called wells. And you put your DNA sample into the well, and then you run a current through the gel. And what's the charge on DNA? Is it negatively charged or positively charged? DNA is negatively charged, right? So negatively charged DNA is going to go toward the positive electrode. And then as it travels through the gel, the DNA separates by size, and the smaller molecules travel farther. You can think of the gel as a bunch of chain link fences, millions of chain link fences that this DNA has to go through. And larger pieces of the DNA can't go through the gel as easily as the short pieces. Think about how difficult it would be for this long stick to be put through 1,000,000 chain link fences compared to putting a toothpick through 1,000,000 chain link fences. It'd be a lot harder to do the big stick, right? So the larger segments of DNA don't travel as far. They might just go a few centimeters. Then the smallest fragments of DNA are going to travel a lot farther. So you compare this to some standard DNA sequences that you already know the size of. Let's get some practice doing this. Take a look at question number 4 in your study guide. Now this is very high yield, about 4 stars. You're probably going to see at least one question like this on your boards. Here we have 8 wells across the top, numbered 1 to 8, and you apply the current, and the negatively charged DNA fragment started moving down from top to bottom. So on the left side of this picture, you see that the bigger, high molecular weight fragments didn't go very far, but the smaller, low molecular weight fragments traveled all the way down to the bottom of the gel. So look at the question. If well 1 contains DNA sample A, and well 2 contains DNA sample B, and well 3 contains DNA sample C, then what can you say about wells 4, 5, 6, 7, and 8? So in well 1, sample A has basically 4 different DNA fragments. You know that because you see 4 bands on the gel under well 1. So there's one that's very small and travels very far, and it has two that are very large, and they don't travel quite as far, and then one sort of in the middle. Then look at well 2, which is sample B. These are pretty long sequences of DNA. None of them traveled very far. Then sample C, over in well 3, has these two very small sequences of DNA that travel very far, all the way down to the bottom of the tray, and one sequence that doesn't travel so far. So what I want you to do is look at wells 4 through 8, compare those against wells 1 to 3, and tell me if sample A, B, or C is found in well 4, 5, 6, 7, or 8, based on how far the DNA has traveled on this gel electrophoresis. So pause the video, take a few seconds to write down your answers, then we'll walk through it together. So what you see here is that on well number 4, this sample has bands that match up exactly with well number 1. So you know that in well number 4, you have sample A, which was in well number 1. There's also a few other segments of DNA that you don't see in wells 1, 2, or 3. So not only does well number 4 have sample A, it also has some other unknown DNA samples that we've not created standard for. Well number 5 is segmenting out like well number 2 did, as well as well number 3. See how those bands all match up? So well number 5 contains both sample B and sample C. Well number 6 has an unknown DNA sample. It doesn't match wells 1, 2, or 3. Well number 7 has sample A and sample B because it looks like a combination of well number 1 and well number 2. And then well 8 has sample A and sample C because it looks like a combination of well number 1 and well number 3. So this is the type of question you might actually encounter on your Step 1 exam. Now look at the next illustration. This is another way you might encounter the same type of problem. And again, you just have to know that small segments of DNA travel farther, large segments of DNA don't travel quite as far in gel electrophoresis. Over here on the right, you have a couple of DNA sequences, and you can see how they're broken into sequences A through E, and you can tell which is the longest and which is the shortest. So take a look at the gel and label each of the bands on the gel A through E based on the size of the DNA sequence. So the sequence that didn't travel very far, the uppermost one, the one closest to the well, is going to be segment B, because B is the biggest sequence. The next one is E, and then A, and then D, and then C is the one that traveled the farthest. That's the smallest sequence. So it's pretty simple. You just have to understand that basic concept. There's also protein electrophoresis, where instead of using a gel to separate fragments of DNA, you create a gel to separate protein particles. So you've amplified the DNA with PCR, and you've separated the fragments by size with electrophoresis. Well, now what? Well, now you can do some blotting techniques. So the first type of blot is a southern blot. A southern blot's the kind of blot they do back home in North Carolina. No. The southern blot was named after a biologist named Sir Edwin Southern, who invented it. Then all the other blots, northern blot, western blot, southwestern blot, those are all just sort of cutesy derivative names. So you have this segment of DNA that you segmented out on gel electrophoresis, and you want to know what it is. Well, you can compare it to a standard that you already have to figure out if this DNA is the same as that DNA over there. So a southern blot is where you take a DNA sample from the electrophoresis gel and you put it on a filter or a cellulose membrane. Then you soak that in a chemical that denatures the DNA so you have separated strands. Then you add a labeled DNA probe that anneals to its complementary strand in your sample. So the probe either has a radioactive label or a fluorescent label. Then you're going to expose that filter to X-ray film, or if it's, you're going to look at the fluorescence, if it's a fluorescent tag. And if it shows up, you know that your probe matches the sample. So if the labeled probe binds, if it anneals, then you know it's the same DNA. You're using a known DNA probe to identify a sample DNA. That's a southern blot, a DNA probe identifying sample DNA. Then a northern blot is very similar, except that instead of your sample being DNA, now your sample is RNA. So you're using a DNA probe to identify an RNA sample. So southern is looking at DNA, northern is looking at RNA. Again, that's your sample. I want you to write down next to that what the probe is. So for the southern blot, your sample is DNA, your probe is DNA. With the northern blot, your sample is RNA, and again, your probe is DNA. Now, with a western blot, your sample is a protein, and your probe is an antibody. So you're using a labeled antibody probe to identify a protein sample. A labeled antibody is used to bind to the relevant protein. So if you're looking for a very specific protein, and you have a predetermined antibody that matches it, you'd be doing a western blot. Now, as a quick mnemonic for what kind of samples each blot uses, a student from South Alabama sent in this. For southern blots, I want you to think down south. And the D in down south should make you think of D and A. For northern blots, think about rude Yankees, and that R is for RNA. And then for western blots, think about cattle ranches out west. So west is where we get beef, and beef is protein. Then there's one more blot called a southwestern blot, which is where you're trying to identify transcription factors and other types of DNA binding proteins. So the probe is oligonucleotides, and the sample are the DNA binding proteins. So if you wanted to determine gene expression, which of these blots would you use? Well, if the gene is being translated and then you're going to look at that protein, you could use a western blot. But if you just wanted to look at the RNA sequence that was transcribed, you would use a northern blot. So basically it all boils down to knowing what the sample is and knowing what the probe is for each of those blotting procedures. If you know that much, you're golden for blots. The purpose of all these blotting techniques is to be able to see DNA. You obviously can't see it with the naked eye, so you have to do something that's going to show up on an X-ray film, or it's going to fluoresce or whatever. Another technique that allows us to visualize what we're working with is called the ELISA test, or enzyme-linked immunosorbent assay. ELISA is looking at antigen-antibody interactions. Now there are two ways to do an ELISA. The first way is called a direct ELISA, because you're looking directly at the patient's blood to see if a particular antigen is present. So you have a microtiter plate with a bunch of wells in it, and affixed to the surface of the wells are antibodies against the antigen you're looking for. Let's call it Lewis virus. So the wells are coated with antibodies against the Lewis virus antigen. Then you add a sample of the patient's blood, and if the patient is infected with the Lewis virus, and there are virus antigens in the blood, or the biopsy tissue or whatever, those Lewis virus antigens are going to bind to the antibodies in the well. Then you add a solution of special anti-Lewis antibodies that have been coupled to a special enzyme that produces some color. And you let those anti-Lewis antibodies bind to the Lewis antigens from the patient's blood. So if the patient has the Lewis antigen in his or her blood, when you add the enzyme substrate, that bound enzyme will cause some color change. So you're able to visualize something that couldn't be seen before. The other way to do the ELISA test is an indirect ELISA, where instead of testing for the antigen in the patient's blood, you're testing for whether or not the patient has antibodies. So in the indirect ELISA, the wells are coated with the antigen, and then you add the patient's blood, and you let all the patient's anti-Lewis antibodies bind to the Lewis antigens in the well. Then you add some antibodies against those anti-Lewis antibodies. And again, if there's a color change, that indicates that the patient's blood contains the anti-Lewis antibodies that you were testing for. So in a direct ELISA, you're using a known antibody to test for antigens in the patient's blood. And in an indirect ELISA, you're using a known antigen to test for antibodies in the patient's blood. The screening test for HIV is a good example of an indirect ELISA test. You're using an HIV antigen to test for the presence of antibodies against HIV antigens. And if that ELISA screening test comes back positive, what's the confirmatory test? A Western blot. Then another visualization technique is called FISH, fluorescence in situ hybridization. So instead of the color-changing enzyme, now you're using a fluorescent probe. And it's not an antibody probe, it's a DNA or RNA probe that fluoresces. So you have a known segment of DNA or RNA that binds to a specific gene of interest, and then it fluoresces because your tagged probe is designed to fluoresce. So again, it's just a way to visualize something. So the fluorescence indicates the gene is present. If there's no fluorescence, that gene's been deleted. Another genetic procedure is cloning. And we're not talking about Star Wars or Jurassic Park. We're talking about cloning recombinant DNA molecules. So basically, you're taking a piece of mRNA that corresponds to the gene you want to clone, and you're going to use reverse transcriptase to turn that RNA sequence into DNA, which we call cDNA or complementary DNA. Then you insert that cDNA into a bacterial plasmid and let the bacteria propagate and replicate, so you can have lots and lots of copies of that cDNA. And because that cDNA was made from the mRNA, it's not identical to the normal gene. It only contains the coding portion of the gene, only the exons. Now let's shift gears a little bit and talk about genetic engineering and making modifications to gene expression. We're still talking about lab techniques, but instead of analyzing DNA sequences, now we're actually manipulating the genetic code of an experimental animal, like a mouse. So you can either delete a certain gene from the mouse's genome, and that's called a knockout mouse because you knocked that gene out, or you can insert genes into the mouse's genome, and that's called a knock-in mouse. Now if you insert the gene randomly, it's called a constitutive insertion. But if you do a more targeted insertion, putting the gene exactly where you want it because it's structurally homologous to the native mouse gene, that's called a conditional insertion. So basically just know those terms, knock in and knock out, and know what they mean. Otherwise, this is a fairly low-yield topic. Then one last topic is karyotyping. you know what this is. This is where you get those metaphase chromosomes, where the DNA is all nice and condensed and organized, and you sort the chromosomes in pairs according to size and shape and the length of the arms and the banding patterns that show up when you stain them. And why do we do karyotyping? Well, mainly to diagnose chromosomal imbalances like trisomies or sex chromosome disorders. So with karyotyping, you're actually physically looking at the chromosome under the microscope to see if there's any big, obvious change in the chromosome. You're not looking at individual particles of DNA, you're looking at the entire chromosome. So on step 1, they may describe a patient with trisomy 21 and ask you what genetic tests could have been done in utero, maybe using amniotic fluid, in order to make this diagnosis. And the answer here would be the karyotype, because you'd see 3 copies of chromosome 21 instead of the usual 2 copies. All right, that's it for genetics. Let's go ahead and do the end of session quiz. First question, what's the difference between southern blot, northern blot, and western blot? With the southern blot, the sample is DNA and the probe is DNA. With northern blot, the sample is RNA and the probe is DNA. And with the western blot, the sample is a protein and the probe is an antibody. Next, what type of test uses a known antigen to discern the presence of an antibody? That's the ELISA test, specifically an indirect ELISA. And then, what type of test is performed in order to diagnose chromosomal imbalances? That's karyotyping. So that's it for Biochem 6. Now, if you're watching these videos in the recommended order, you've made it about 2/3 of the way through the Part 2 videos. So let's take just a minute to think about how to approach the last few days of your study process. First of all, if you finish going through the Part 2 videos, and say you have a week or so before your test day, you might take your main review book with all the annotations you've been making and divide the number of pages by 5 days and dedicate yourself to reading that number of pages every day. So you're going to get through the whole book one last time over the course of 5 days. And as you do this, keep a list of small details that maybe you've overlooked or that you don't feel like you've mastered. Then review that list at the end of each day and at the beginning of the next day. That process is going to ensure that you've read everything one last time in the last 5 days before your test. You should also go back through your DIT study guide and review the rapid fire facts and anything else that you noted as being particularly important to remember. Then, on the last day before the test, relax a little bit, especially in the afternoon and the evening. Get a little exercise, eat a nice healthy dinner, get a good night's sleep. Make sure you have your ID and your USMLE admission pass, and you have directions to the testing center, and it's all gathered up so you're not scrambling around looking for all that stuff on the test day. Make sure you set your alarm, or 2 or 3 alarms. There's plenty of time for a relaxed morning and a good breakfast. You want to dress comfortably, not just in terms of whether the clothes are comfortable, but also, are you going to be self-conscious or distracted thinking about whether or not people are looking at you? If you feel most comfortable being a little dressed up, go with it. You want to pack a lunch and some snacks. You want to bring a jacket or a sweatshirt in case the testing center is kind of cool. And then finally, we put together a short list of some things we think you might want to put on your marker board at the beginning of your first block. That's entirely up to you, but if there are things that you know you're likely to get hung up on, write them down. All right, that's the end of this genetics portion of Biochem. Next up is psychiatry.